Good afternoon, everybody, and welcome to the Carline Cymru revision session. This session will focus on defence from pathogens and the use of vaccinations for GCSE biology, and will be presented by Mrs. Sedat from Bishop Headley in Merthyr. The session will last around 45 minutes, where the teacher will go through the relevant subject content. If you have any questions, please use the question and answer section, and we will endeavour to answer your question during the session. The session will be recorded with the recording and any relevant resources uploaded to the e-sgol website in the Carline Cymru area. Thank you.
Good afternoon, everybody, and welcome to the third biology session for this season. This session is on defence from pathogens and the use of vaccinations, and this will be a two-part presentation, one this week and then the rest of it next week. So let's have a little look at what we're going to focus on in today's session. So the first thing I want to look at then is what a pathogen is, and for you to be able to name the 4 different pathogens that we're going to look at today. We're going to look at the first line defence, so how the body actually protects us from pathogens getting inside the body, so what features that we have in order to stop them coming in the first place. We're then going to look at our second line defence, so if those pathogens do get inside our body, what does our immune system do about it? And we're going to look at 2 very distinct different types of white blood cells today called phagocytes and lymphocytes. Then we're going to look at the difference between primary and secondary response to a pathogen. So if we get ill the first time, and this is the first time we've ever had any response to that pathogen, what does our body do to respond? And then if we were to be exposed to the same pathogen a second time, how is it different? And finally, we're going to look at how vaccinations work, what is a vaccine, and how they actually work to protect us. Okay, so let's get started. The first thing I want you to consider is this question: are all microorganisms bad? Now we know that microorganisms are everywhere. We are exposed to microorganisms whenever we do anything, whenever we eat food, whenever we go outside in the air, anything that we do, we are being exposed to microorganisms. And here is a definition here of what a microorganism is. So a microorganism or microbe is an organism of microscopic size, meaning that we'd need a microscope in order to see it. Most of the microorganisms that we come into contact with cause us no harm, and some of them are even helpful. Okay, so for example, they love in our guts. No, they don't. They live in our guts and they help us with digestion. However, there are some microorganisms that can cause harm, and we call those pathogens. So in the exam, you need to know that word pathogen. You need to know that a pathogen is a microorganism that would cause us some harm. And there are 4 main types of pathogens that you need to be aware of. The first one is a fungi. Okay, and I've got an example there in a diagram of what a fungi might look like. So this one here is athlete's foot. That would be an example of a fungi. Protists, so diseases like malaria are caused by protists, which are just single-celled organisms. Bacteria, such as salmonella, or the bacteria that causes food poisoning from things like uncooked chicken, that would be an example of a pathogen. And finally, viruses. Obviously, we've been exposed to a virus quite a lot in the last 2 years, and there's an example there is COVID-19. So how do pathogens actually make us ill? There are 2 ways that pathogens make you ill. The first one is that they release toxins. Now, toxins are a poison that can cause damage to the body. So toxins are harmful substances produced by bacteria or other pathogens that can poison body tissues and enzymes, causing damage. So there's a little diagram here at the top where we're going to come back to later on. Okay, so we can see here I've just really simplified this microorganism, and I've just used a simple shape, and we can see here that all the X's are toxins, and if these toxins are released into our blood or into our cells, then they can do damage to our body. The second thing is shown by the animation at the bottom, and you can see here that this is because they reproduce. So here we've got bacteria, In one of our sessions, we looked at how bacteria reproduce. They reproduce by a process called mitosis, which is binary fission. And this is that every bacteria then will duplicate into 2, and 2 will go into 4, and 4 will go into 8, and so on. And in the right conditions, so that be warm and moist, and making sure that they've got enough nutrients, they're able to reproduce at quite a fast rate. Now, this rise in the number of pathogens that we can see in the animation can cause damage to a cell. And if the reproduction rate is so high, it can cause a cell to burst. Okay, and this is that the pathogens themselves, they take away the resources that a cell will need to survive. So both of these things, the fact that they produce toxins, which are harmful substances that damage our cells, and the fact that they reproduce at a fast rate is 2 of the ways that pathogens can actually make us ill. How do microorganisms enter the body? Have a look at this picture here of this man. Have a little think to yourselves, how would they actually get into our body in the first place? So have a little think, say it out loud, pause the video if you're watching this offline, and have a little go of this, thinking about how they may get into our body. Okay, so if you've had a chance to pause the video, then you can do so. Let's have a little look. So number 1 then, into our eyes. Okay, so we've got to think that microorganisms can get into the body anywhere where there is not skin cover. So that could be into our eyes, in through our nose, in through our mouth. If we have any cuts on our skin and they haven't scabbed over, then in through our cuts and in through our genitals. Okay, so anywhere where there is less skin, okay, that's where microorganisms can enter the body. And we're going to look now at how the body actually protects itself from microorganisms trying to get inside into our blood. So we call this our first line defense. Okay, so we can see the pictures at the bottom there, start to have an idea about how our body tries to prevent microorganisms from getting into the body. So our first line defense is a way that the body tries to protect ourselves before our immune system has to kick in. So looking at the pictures at the bottom then, okay, we've got the first one, we've already discussed it. This is our skin. Now, if we think about our skin, okay, I'm sure everybody has had a cut or a graze on their skin. The cut or graze will immediately start to heal itself. Okay, so platelets, which you looked at in year 10, will start to accumulate in the site, and then eventually they will form a scab. Now, the skin itself is known as a physical barrier. It's one way that the body tries to stop microorganisms from getting into our body in the first place. Second one then, as we know, the eyes is one way that they can get into our body. If we can look at the picture there, okay, our eyes, if anything actually get into our eyes start to water, and that is our eyes actually producing tears. Now, tears contain an enzyme, and that enzyme then can try and prevent bacterial growth from happening inside our eyes. Because it contains enzymes, it's no longer a physical barrier, but instead now a chemical barrier. Okay, so if those microorganisms get inside, they can cause a chemical barrier. The enzymes inside our tears try and prevent from bacterial growth. The tears themselves are quite salty, and bacteria don't like salty conditions. So that's another way that our eyes try and protect us from microorganisms from getting inside the body. Another way that microorganisms get inside the body is through our nose. Okay, so if we have a look up the nose, okay, we can see that the nose is lined with hairs. So we have hairs that are inside of the nose, and what they try and do is they try and capture any microorganisms that try and get into our nose. Now, the hairs can only do so much, okay, and instead we actually have cells in our nose that produce mucus. Now, the mucus itself is there to trap pathogens. And if we think about year 10, when we looked at the respiratory system, we have tiny hairs that we're going to look at in a moment that also do the same job. Okay, so the mucus itself tries to trap any microbes or microorganisms that get inside our nose. Okay, sticking with our first line defense then, some other ways. So the picture at the bottom then, if they've got past our nose or they've got into our into our respiratory system, so into our trachea and our bronchi. Then the cells that line the trachea or the bronchi have these tiny microscopic hairs called cilia. Now the job of these hairs is to move mucus up to the top of the throat. So we have two types of cells. We have ciliated epithelial cells, which are the cells that contain cilia, and their job is to sweep mucus. And we also have goblet cells that line our respiratory system, and their job is to create mucus. The mucus is there to trap the microorganisms, and the cilia is there to actually sweep it up to the top of the throat. Now, if we eat something that has bacteria on it or has pathogens on it, then our stomach has ways to try and kill those pathogens. And as we can see from the diagram here, it contains an acid, a hydrochloric acid. Hydrochloric acid is very strong, okay, so it's strong enough to kill any pathogens that have been consumed in food, or if we link the two together, that have been caught up in the mucus that has now been swallowed into the stomach. Okay, so this is our first line defense, and you would be required in the exam to be able to recall some of this knowledge. Okay, so let's just go back over it again. So to prevent it from getting into the body in the first place, we have a physical barrier called the skin. We have tears that contain enzymes known as chemical barriers. We have mucus and hairs in our nose. We have cilia that run through the trachea and the bronchi in our respiratory system, and we have acid in our stomach just in case any of those pathogens get in. Now, what happens if pathogens do get inside of our body? Well, if they get past our first line defense, remember our skin and our hairs in our nose and our tears, all of that is considered our first line defense, then they could cause an infection. Okay, and this is where our immune system, you may have heard of that term before, starts to come in then to try and minimize the infection that can come. Now, the immune system consists of two types of white blood cells. So when we looked in year 10 at blood, one of the cells that we looked at was the white blood cell, and we know white blood cells are there part of our immune system to try and fight off infections. And now in year 11, we need to know white blood cells in a little bit more detail. Okay, so instead of just calling them white blood cells, we need to know that they are called phagocytes and lymphocytes. Now, spelling of those words is going to be required, so make sure you stop and you practice the spellings of those words. And these two types of white blood cells have two very different jobs. Now, the first one that we're going to look at then is called a phagocyte. Okay, so I've got a little animation there of what actually is happening. Okay, so this structure itself here, which I'm just showing you, this is the phagocyte, and then what you can see going inside is the pathogen. Okay, so just to make sense of this video, okay, so the purpley abstract shape thing here is the phagocyte, and then the thing going inside is the pathogen. So the first thing that happens then is that the phagocytes are attracted to pathogens in the blood. Okay, so the first thing that happens is they're attracted to them, so they hunt them out, and the first thing then is they bind to them. Okay, so we can start to see that it's starting to bind just at the beginning of the video. The phagocyte's membrane then, so remember it's surrounded by a cell membrane, starts to surround the pathogen and it starts to engulf it. Now the word engulf just means to take it inside. Okay, it's a really useful word to learn for the exam because sometimes students struggle to describe what actually happens. Okay, so we can see, watch it again, we can see the membrane of the phagocyte starting to surround the pathogen, and then the pathogen then actually moves inside. Okay, that is the phagocyte engulfing it, okay, taking it inside. Now once it's inside, the phagocyte itself has digestive enzymes, and these enzymes, they break down the pathogen in order to destroy it. Okay, so let's go through it again. The phagocyte is attracted to the pathogen and it binds to it. The pathogen then starts to go inside, so the phagocyte engulfs it inside of it, and then enzymes inside of the phagocyte then start to break it down and destroy it. So that's the function of phagocytes. Now, lymphocytes are slightly different. Okay, so lymphocytes have a very specific job. If I explain this animation here, okay, I'm going to talk you through in steps. So we got a little flu virus here, okay, and the flu virus, as you can see, is in the blood, and a lymphocyte starts to come along, and it recognizes these very special proteins that line the surface of the pathogen. Now, these very special proteins are called antigens, okay, and you need to know that word for the exam. And each pathogen, whether it be a virus and a bacteria, they have distinct shaped antigens on the surface. So lymphocytes, they come along and they start to recognize a particular shape of a protein on the surface of the pathogen, and we call those antigens. The next thing that happens then, as we can see here, we've got the lymphocyte then producing something called antibodies. Now, the lymphocyte itself will recognize from the first video, okay, so let's see it again. It will recognize from the first video that that flu virus doesn't belong in the body. We say that it is foreign or not naturally occurring within the body. Okay, so the lymphocyte itself, once it's located it and it looks at the proteins, the antigens on the outside of the flu virus, it will realize that it doesn't belong there. And as a result, it will start to produce these structures called antibodies. Okay, there's specific proteins called antibodies, and we can see here that the lymphocyte is producing lots of them. Now, it takes lymphocytes a little while to start to make a very specific antibody for that specific pathogen. The lymphocytes then release these antibodies, okay, releases the antibodies into the blood, and if we watch this part of the video, the antibodies themselves start to stick to the special antigens on the outside of the virus, and they start to make them stick together. So here we go, we've got 3 viruses now, okay, the antibodies, which are these little Y-shaped structures, they start to make the pathogen stick together so that the phagocytes can come along then and engulf them in one, okay, so it makes it more effective. Now, that was a lot to learn in 3 animations there, so I've got a little picture just to kind of explain that again. Okay, so here we've got lymphocyte 1, and we can see this lymphocyte, this white blood cell then, has recognized, here it is, this is the pathogen here, the one that I'm putting a shape around now. This is the pathogen, so it recognized the pathogen, and it's noticed that it's got specific antigen shapes on the outside of the surface of the pathogen. Okay, so this is a virus or a bacteria or a fungi, and they have these antigens, these very special proteins on the outside of the surface. The lymphocyte will come along, it will recognize them, and then it will start to produce antibodies. These are the Y-shaped structures here that we can see. These are the antibodies that will fit the specific shape of the antigen. Now, it says here antibodies which have a complementary, so a specific shape to the antigens on the pathogen. Okay, so this is the lymphocyte here, which I'm drawing an arrow to. This is the pathogen, okay, that I've drawn a circle around, and these are the antigens that are being made. The antibodies then, as you can see, have a very shape. And these will then cause the pathogens to stick together so that a phagocyte can come along and engulf them in one. Okay, so you can see here from these two are producing different shaped antibodies. Each pathogen will have a unique shaped antigen, those special proteins on the outside of its surface. So the lymphocyte has to be able to produce different shaped antibodies that are going to fit the antigens on the surface. And as we can see here in the 3rd diagram then, we're causing them to clump together. This is them clumping together because of the antibodies now. The antibodies are causing them to clump together, and then what will happen, this is the phagocyte here that I'm drawing a box around. And the phagocyte then will come along and it will engulf them as one. So it makes the process more effective. Okay, What else do lymphocytes do? So we know, number one, that they produce antibodies. Okay, they produce antibodies for the antigens. They also produce antitoxins. One of the things that we mentioned right at the beginning of the video is that pathogens, we saw this bit of the picture, pathogens or microorganisms, they create something called toxins. Now, if they create toxins, this is what's going to make us unwell. We need white blood cells, the lymphocytes, to produce something called antitoxins. Now, the word anti means opposite, the opposite. So if you've got toxins, then the antitoxin is going to neutralize the toxin. Okay, so it's going to make sure that the toxin doesn't cause us to damage the tissues. So lymphocytes produce antibodies and they produce antitoxins. Okay, so we have lots of words now that we've got to put together. We've got antigens, we've got antibodies, and we've got antitoxins. So make sure that you don't confuse those words when you get into your GCSE exam. So here's a summary then of the 3 ways that white blood cells fight off pathogens. Okay, and I've put them into text so that you can then summarize these on a flashcard. Okay, so one of the things that they do is they release antitoxins. So white blood cells, whether it be lymphocytes or phagocytes. So have a little think now. Do lymphocytes or phagocytes produce antitoxins? Okay, as we can see here, they are lymphocytes. Lymphocytes release antitoxins. Okay, and their job is to bind to the toxin and neutralize its effects. Okay, so what I've tried to do is I've tried to summarize now in just information that you put in a flashcard and that you'd learn off by heart. Okay, the second thing they do then is they release antibodies. Again, think to yourself, is that a lymphocyte or a phagocyte? These are also lymphocytes. Okay, so lymphocytes then release antibodies. Okay, and they bind to antigens. Antigens are on the pathogen. Antibodies are released by lymphocytes. Why do they do that? To make them clump together so that they can be engulfed easier. And the last one then is that they ingest pathogens. Okay, so is it lymphocytes or phagocytes that do that? This time it is the phagocyte. Okay, so this is the phagocyte, and their job is to ingest, consume the pathogen. Okay, they engulf it and then they destroy it with digestive enzymes. Okay, so there are 3 that you need to know, 3 functions of white blood cells and how they fight off pathogens. I would say put these on a flashcard and learn them off by heart. Okay, so here are 3 quick questions. Okay, these are the types of questions that you may be asked in the exam. I've allocated some marks for them. Okay, so the first question is, how do pathogens make us ill? And that would maybe be a 2-mark question because you could say give 2 reasons why. Question 2, state 3 ways white blood cells fight pathogens that enter our body. And that could be a 3-mark question. And then the last one then, 4-mark question, describe how antibodies fight pathogens. Now, if you're watching this video offline, then you would be able to pause this video at this point, have a go at the questions, and then the answers would follow. So I'm just going to wait 20 seconds now to have a little think about one of these questions. What would you write? And then I'm going to go through the answers. Okay, so let's have a little look at these questions then. So number 1 then, first question was, how do pathogens make us ill? So pathogens make us ill by releasing toxins. That would be the first mark. Okay, they release toxins. And then the second one then was they cause cell damage by reproducing. Okay, so 2 marks there. Have a look, how many marks would you have awarded yourself? Okay, second question then. State 3 ways white blood cells fight pathogens that enter our body. So the first thing that we say then is that they release antitoxins. So remember, they are the lymphocytes release antitoxins. The second one then is they release antibodies, also the lymphocytes. And the final one then is that they ingest pathogens, and those are the phagocytes. So 3 marks available, 3 things that white blood cells do in order to fight pathogens. The last question then, describe how antibodies fight pathogens. Now this would be a much more detailed answer, okay, so we can see that antibodies are released in response to a pathogen. The antibodies are specific, okay, so we need to know that they are a specific shape and that they can bind to only that pathogen. The antibodies label the pathogens and cause them to clump together, and then finally the pathogen can be destroyed by the phagocyte actually engulfing it. Okay, so much more detailed answer there. That's the sort of level that you want to get to then in terms of your knowledge about how white blood cells fight off pathogens. Okay. The next thing we're going to look at then is memory cells. Now, white blood cells form memory cells, and they kind of do what they say on the tin. Okay, they are our cell's memory, so that if we've been infected with an infection with a type of pathogen once before, then these memory cells stick around in our body, and hopefully they avoid us getting too ill a second time when we have an infected by a similar or same pathogen. So this means they remember how to make the same antibodies, okay, if the same pathogen ever gets inside the body. Okay, so if we potentially have had tonsillitis, okay, so we've had tonsillitis before, so we've made the antibodies that would respond to tonsillitis. If we have that exact same pathogen enter our body again, then our memory cells would remember how to make those antibodies. Now, this allows the immune system to respond much more quickly. Now, see how it doesn't say that you won't get that illness again. You will still get the illness, but it means that hopefully your body will be able to respond quickly so that your immune system then means it can fight off the infection before you get too ill, before too many toxins are released, and before there's too much reproduction of the pathogen. Okay, here's an example of a foundation tier question. It says the following statements describe how the body responds when it is invaded by pathogens. The statements are not in the correct order. So number 1 is the pathogens have foreign antigens. Step 2, pathogens enter the blood. Step 3, they respond by releasing antibodies to the antigens. 4, the antibodies attach to the pathogen. 5, the antigens are recognised as foreign by lymphocytes, and 6, the attached antibodies help phagocytes destroy the pathogen. 3-mark question, 2 of the steps are already done for you. Have a little think now, in what order would you place these statements? So what's the first statement? Have a little look. The first thing that happens then is that the pathogens have got to enter our blood in the first place. Okay, so step 2 is the first one, pathogens enter our blood. Then we know that it's step 1, so the pathogens have foreign antigens. What would be the next step? 5, you're correct. The antigens then are recognised as foreign by lymphocytes. Then they respond by releasing antibodies to the antigens. And then what would be the last 2 steps? The antibodies attach to the pathogen, and then the attached antibodies help phagocytes destroy the pathogen. So the order was 2, 1, 5, 3, 4, and 6. Okay, so it's a 3-mark question, so you would have needed them in order for the 3 marks. Let's have a little look now at primary and secondary immune response. Okay, primary, the first time you're exposed to a pathogen. Secondary, the second time you're exposed to a pathogen. And often you will see this graph in an exam. Okay, so you may have seen it in your lessons if you've already studied this topic already in class. Okay, this is a very common graph. This is the, as we can see, we've got time along the bottom. Okay, so time in terms of exposure and response. So time in terms of exposure and response. And then at the side then on the y-axis we've got concentration of antibodies. Okay, so this is the concentration of the amount of antibodies that would be in our blood. Now, as we can see, if we are exposed to a pathogen the first time, which is the initial exposure, so the first exposure, it takes quite a long time for the lymphocytes to produce the correctly shaped antibodies. Okay, so remember, they haven't seen this pathogen before. They've got a very specific shape on the edge of their structure called antigens, and it's going to take our lymphocytes a little while to start to work out what shaped antibodies they need to produce. So we call this the primary immune response. And as you can see, it takes a while. Okay, so it takes quite a while for the concentration to rise, and the final concentration isn't very high at all. Okay, so this is why if we're exposed to a condition for the very first time, we get quite ill, because it takes a long time for the lymphocytes to actually fight off that infection, produce the correctly shaped antibodies, and therefore get rid of the bacteria, virus, or fungi that's causing it. Let's have a look at the secondary exposure. So this would be very unfortunate if you had it the second time straight away, but the second time your body is exposed to a pathogen, have a look at this response. So you can see that it takes the lymphocytes much less time, so we can see a very steep line on the graph. It takes them not very much time at all to actually produce a huge amount of antibodies, and this is called the secondary immune response. The reason why it doesn't take the body very long, and we are able to produce a very high concentration of antibodies in a short space of time, is because of memory cells. These memory cells remain in our body, and they are able to remember the shape of the antigens and therefore be able to produce the correctly shaped antibody. And this is why our secondary immune response is much steeper, can produce a high number of antibodies in a very short space of time. So hopefully the second time you get an illness, you don't suffer with it as much. So let's have a little look at this. We got a faster response, and the number of antibodies produced is larger. So if you were asked to analyse how is the initial exposure or the primary immune response different to the secondary immune response, you would say the secondary immune response is faster. So that would be the first one, and the number of antibodies produced is larger. Okay, so the concentration is higher. Okay, so it's a faster response, and we're producing a larger number of antibodies. Okay, so here's two other questions. Okay, so have a look at these. Question 1, describe what happens during the primary response to a pathogen. And then question 2, describe what happens during the secondary response to a pathogen. So both of these would be a 2-mark question. There are some keywords at the bottom. So you would pause the video at this point, have a go at these questions, and then we would then mark. Okay. So here are our answers. So the primary response, when a pathogen first gets into our body, it takes the white blood cells or lymphocytes a little while to make the right antibodies, or you could say longer. It takes them longer to make the right antibodies. This is known as lag time. The antibodies are made and the pathogen is destroyed. That's our primary response, the very first time that we're infected with a condition, with a pathogen. Number 2, then, the second time the same pathogen gets in, the response is much quicker. So this is our secondary response. More antibodies are made, and this is because memory cells have remembered how to make the right antibodies. Okay, so have a look at those 2 responses. You would need to be able to answer both of those questions there. Okay, right, we've got 10 minutes to go, so let's have a little look at what a vaccine is. Okay, so in your exam, whether you're sitting foundation or higher, you need to be able to write a definition of what is in a vaccine. This is the vaccine here, okay? This is the vaccine. And as you can see here is the COVID-19 vaccine. Some of you may have had it. When you go to receive a vaccine, what's actually happening is that they are actually injecting you with a dead or inactive or weakened form of the pathogen. Okay, so COVID-19 is caused by a virus. So what they would be injecting you with then is a dead, inactive or weakened version of the COVID-19 vaccine itself. Now the vaccine itself, or the pathogen, would contain the antigens that the body has to respond to. So what we're doing is we're actually creating the primary response that we've just seen in the graph below. Okay. So let's have a look at how they actually work. Again, I've tried to summarize this in steps, okay? So step 1, the vaccine is injected into the body, and what we're injecting then is a dead or inactive version of the exact pathogen that causes that disease, okay? So here are the, let's say that this is a bacteria, and we can see the antigens that are on top of it. So we actually inject ourselves with a dead or weakened form of the pathogen. Step 2, the white blood cells or lymphocytes recognize the antigens on the pathogen don't belong to the body. So this is exactly the same as the response that we had before. So this is our primary response in the fact that lymphocytes will then make complementary antibodies to fit this structure. Okay, so the antibody specific for this structure would then be made by the lymphocytes itself, and the body would respond just as it would with an infection of any other pathogen. Step 4 then, okay, in our blood then we keep behind our memory cells, so our memory cells remain, and if we are then if we are then infected by the same pathogen again, then we would see the secondary response happen. Okay, so if the same pathogen was enter our body, this is what we would see. We should then see a secondary immune response. Okay, and this means that we are immune. Now, lots of students get a little confused with the idea of immunity, meaning that we're never going to get that disease. So just because we've had the COVID-19 vaccine doesn't mean that we are not going to get COVID-19. It just means that if we were to be infected with COVID-19, our white blood cells would be able to produce the antibodies quicker. So therefore, we can make more of them so that the pathogen is destroyed before you feel the symptoms, or you feel the symptoms so very badly. Okay, so being immune does not mean that you're not going to get that disease. Okay, it just means that if you are infected, then you will start here on the secondary immune response rather than here on the primary immune response. Okay, so vaccines are preparing our bodies just in case we are infected with that particular pathogen. Okay, so why do we sometimes have to have numerous vaccines like the COVID-19 vaccine or even the flu vaccine? Now, some of you may have a condition that requires you to have the flu vaccine every single year. Okay, if you're one of those people, then the reason for that is because the virus that causes it, influenza, its DNA mutates frequently. So because its DNA mutates, we almost get a new flu virus every single year. Okay, there are slight tweaks and changes to it. Okay, and because of that, there are new antigens produced. So even though our white blood cells, our memory cells, are left behind and they know they've been infected with flu before, it could be that this year we have a slightly different variation on the antigens, and therefore our white blood cells, our memory cells, lymphocytes, they do not know how to respond. So the memory cells will not recognize this year's new strain of flu, and so a different vaccination is needed to give immunity for the year. Okay, so that's a question that sometimes comes at the end of a bigger question. Why do some vaccines need to be given every single year? And you would say because DNA mutates, so therefore it would have different antigens. Okay, the memory cells would not be able to respond. Okay, we've only got 5 minutes left. I'm very conscious of time. So let's have a little look at some past paper questions. Whatever I don't get to complete today, the PowerPoint will be available for you, so you can go over them at your own leisure. Okay, so this is looking at a very specific vaccination. So immunity against polio is created by a series of vaccinations. Now, you've probably all had the polio vaccinations, and the reason why you probably can't remember is because they're given at age 3 months. Okay, and then they're given at a range of ages then, and we call those booster vaccinations. Okay, now this graph here, as you can see, we're given the first vaccination at 3 months, we're given our first booster vaccination at 4 years, and then we're given our second booster vaccination at 15 years. Now this question says, use the graph and your knowledge, so use the graph and knowledge to state the age range during which a person is most likely to suffer from polio. Have a little think about what you would do for this question. So remember, you're going to most likely suffer from this polio when you're unvaccinated, okay? So it would be, the answer will come up. What's going on here with my PowerPoint? There we go. 0 to 1 years, okay? So before the vaccination is given, that's when, as you can see, the level of antibodies in the blood are very low, okay? So this is where we are most likely to suffer from polio. The second question then says, describe how the immune system reacts to booster vaccinations. So this is how the immune system reacts to booster. This is a 5-mark question. This is from the old legacy. So let's have a little look at what you'd have to say for 5 marks here. Number 1, we have to mention the ideas that we have memory cells, okay? And these memory cells remain in our blood. Once we have memory cells, the job of the memory cells is to recognize the antigens of the dead or inactive pathogen that we're being injected with. Okay, so how do we react to a booster vaccination? They recognize the antigens of the dead or inactive pathogen that we've got now in our booster vaccination. This then triggers white blood cells or lymphocytes to form clones or reproduce or multiply, which then produce antibodies more quickly. So the way that we produce lots of antibodies is by those lymphocytes replicating themselves, reproducing, creating clones, multiplying into vast numbers so that our bodies fight it off quicker. Okay, so that would be a 5-mark question there. I'm going wrong with my PowerPoint here. Okay, here we go. Is another question then. The diagram below shows stages of the response by one type of white blood cell that causes a flu virus vaccination. So we've got 3 stages. So we can see the flu virus being injected. Stage 1, flu virus antigen is injected into the blood vessel. Stage 2, the antigen becomes attached to antibodies on the membrane of the white blood cell. And then stage 3 doesn't have, I haven't cut anything off there, it had nothing, had no information. Now the question is, part 1, name the type of white blood cell that produces antibodies. And this is a 1-mark question. So have a little think what you would write for that one. Oops. Here we go. So we've got lymphocytes. Describe the process that has taken place between stages 2 and 3. Okay, well, stage 2 is here and stage 3 is at the bottom. And as you can see, antibodies are being produced. Okay. And then, oh, I think these are on the wrong order, actually. Okay, I think this one should describe the process that's taking place between stage 2 and 3. I think it should be this one, sorry. I'll change the PowerPoint ready for when you have it on the classroom, okay? And then the last question then, state the importance of stage 3 in protecting the body, antibodies produced, so there will be a rapid response. These have just come up in the wrong order. I apologise about that. There's one more question, I think, which is a 4-mark question, similar to what we looked at before, so you can practice this one at home when you get 2 minutes, okay? So pause the video at this point, have a go at the question, and then the answer will come up after. Okay, so the session is nearly over. What I'd like to do is thank you for taking part in the revision session today. What I'll make sure I'll do is I'll change anything that's wrong on this PowerPoint. It'll be uploaded to the classroom and then you can have a go at these past paper questions then at your own leisure. I'll also make sure that there are the links to the WJC website as well, so you can access all the blended learning activities as well. Okay, thank you so much, everybody. Bye.